hopefully each time if we implement a new methodology, there are some challenges. So always we are taking the questions, how long does it take? What are the pitfalls? What should we do in first? So have we ordered additional reagents from several companies? So before starting, we had a lot to do. And now I will take you with me about our journey to implement the long-read technology. So I have to disclaim that this is our journey. If you begin with long sequencing technology, maybe you learn something from our pitfalls. I will show you, but also if you're already doing long-read sequencing, maybe you keep in mind what you had experienced first, or maybe totally other ones. So first of all, implementing a new methodology, don't panic. So hopefully you have a towel with you. Instead, in my case, I have a t-shirt. So therefore, nothing bad could happen. But we will see. So the potential of long-read sequencing, why we are interested in to implement it. So there is a better de novo assembly. It's not always necessary, but it's nice to have. But also there's an improved detection of the structural variants we will see with short-read technologies. There's also the possibility for detection of repeat expansion. I will show you later. And also there's a more uniform coverage of the CG-rich region, so especially the first exons or the promoter regions. And also there's a possibility for haplotype phasing. So that means that you can detect the heterozygous variants, whether they're in cis or in trans, or the same or on different alleles. As well as, in addition, there's a possibility to have an epigenetic analysis regarding the methylation detection you will get directly during sequencing without any further steps or something like that. Also, this is an advantage. And in total, it offers a more comprehensive diagnostics. So in the past, we had several different methodologies we do in parallel or after each other. And maybe now we have one where we get most of it out or get all of them. So let's start. So in the beginning, we thought, okay, what do we need? We need an instrument. So we went for Oxford Nanopore Technologies and we decided to go for a smaller instrument because we don't want to exchange the short-read technologies we developed over the years. We had several short-read instruments, starting from the MiSeq to the NextSeq to the large NovaSeq with a high flexibility and also with a high capacity. We do thousands of exomes per year and an increasing number of genome sequencing in the short read part, but therefore we decided to go for a smaller instrument. And as always, we do it the scientific way. We do it by our own. So therefore we decided to go for the P2 Solo instead of the P2 Integrated, who also offers a lot of hardware resources. So our IT department said, okay, let's save money. We have CPU computational resources, we have GPU computational resources, you can get them, and then you don't need any hardware. Okay, let's do this. So therefore, we decided to go for the genome sequencing as well as adaptive samplings, and I will show you in a minute. So we also ask us, so what are the different steps we have to make, and what is the time frame we need for the whole pipeline? So therefore, it's clear we have to do the DNA QC, we have to do the library prep, the sequencing itself, and afterwards or in parallel, we have also do the first analysis on the data, so the generation of unaligned BAM files, the secondary. Therefore, we choose the varvis software and later on the interpretation of the results. So for reading the manuals, we get an idea, okay, library prep should be done within 1 day. So it's more or less comparable to the short-read sequences where we have in 1 or 2 days several exomes or genome datasets and can directly start. So the sequencing was somewhat different. So we didn't know. We read a lot about other resources, public forums or something like that. So yeah, maybe around 3 days. So this is in comparison to the short-read taking a long time. So on all short-read instruments we use so far, within 24 hours, you have the data set. Whether it's a small panel, it's a large panel, it's an exome or a genome, within 24 hours. Also, the time needed for the unaligned BAM file generation or base calling is somewhat different, but we will see in a few minutes how this happens. And as I told you, we want to use the varvis software, and also we had a lot of discussion to implement the automated upload and also analysis way with the guys from the company. So the first part was quite easy. So we have to do the QC as normal, so it's routine in our lab for several years to measure the concentration, the integrity of the DNA, as well as get an idea about the fragment length, because also in long read sequence, it's not that long as much as possible. So also there are guidelines from the company that you need a special fragment size for the optimal usage of the flow cell. So within the library prep, it's easy. We have double-stranded DNA. We have to fragment them to the recommendations of the Oxford Nanopore Technology Company for the whole genome sequencing. The fragments should be around 12 to 15 kb. Also the application specialist we talked to give us some hint on this step. And if you use adaptive sampling, so the fragments should be a little bit smaller, so around 8 kb. So after end repair and ligation of the sequence adapters, you directly can start the sequencing. If you use the adaptive sampling, you have to incorporate barcodes, because on one flow cell you have up to 3 samples, but all of them is possible within 1 day. So it's really fast. So we can start in the morning and at the end of the working day. So also keep our technicians in mind, they have an 8-hour day and not a 12 hours for the scientifics or something like that. So in a normal working day, it's possible. So our first results was to keep the DNA input and integrity was easy. So it's standard. So check. So also the fragmentation to the recommended size, also we evaluated this by the tape station analysis, was easy. Check. So we have a run. To get a final amount of at least 1 micrograms, it's much, much more than we need for the short read sequences. Also, therefore, we have to exchange our DNA extraction methods not to the quality, but to the amount. Because in the last year, we reduced already the use of blood or other materials to extract the DNA because it was not necessary to have that much. But now we change this. We need more of the DNA to achieve at least 1 micrograms of the sequencing reading library. But also this was quite easy. So the first step, check, check, check. Yes, we are on the right way. But then, yes, maybe we face some challenges. So let me show the first results of the sequencing itself. So Oxford Nanopore guarantees that each flow cell have at least 5,000 active pores. So that wasn't the case for all of the flow cells we had. So I here show you the minimum, maximum, and the average of the preliminary or values for the sequencing results. So we had flow cells that really achieve hardly this limitation of 5,000 pores. So the first one you can see here with the 4-cell check at more than 5,000, then below the library and starting the sequencing. And the first one we saw at starting the sequencing, the flow cells directly dropped below 5,000. So we expect that the results from this flow cell will not achieve a 30x coverage for the whole genome, but let it run. So it's one of the first experiences and we don't want to stop them. So, but we had also very high quality flow cells with more than 9,000 pores, and in average, we get more than 8,000. Also the sequencing time with this low quality flow cell, the sequencing ends shortly after 2 days. But we also have flow cells which ended only after 4 days. It's nice to have because the output is much higher. Also the coverage at the end will be much higher. And if you can see here, the output for this low quality flow cells was 40 gigabases. It's too less for genome sequencing, but we also have a high performance flow cell with here nearly 200 gigabases of data. And in average, so around 120 or so should be enough for a 30x coverage. And as you can see here in the maximum and also in the average, we reached most of them, of the flow cells we used at the end, a necessary coverage for genome analysis. So all these values are from this outliner, really poor quality flow cells. But I have also to state that so far we are doing this long read sequences for 2 months. We had a lot of flow cells used, as much as possible in this time. I will show you in the next slide. But half of them were of poor quality or in the flow cell check never reached the 5,000 pores. So maybe we are the lucky guys, we get the bad patch. So I don't know, and this is our experience. It's not a main issue for all the other users. I don't know. I talked to a few, but maybe they don't say them, but we are the secret rule. So therefore I present these results. But I have also to state that it was no problem to get a replacement by Oxford Nanopore Technologies. So if we check in first that the flow cell will never reach the quality limitations and take the other one, so it was directly replaced within a few days. So it was no disadvantage at all, but we also will keep on an eye on that in future. So let me say something about the IT hardware. So IT department tell us in the first line, yes, you can have all the resources you need. And at the time I need them, they say, oh, no, not yet. There's an AI experiment. It's very important. You can have them. It's okay. Then we have to do this otherwise. So therefore, we are not on the high computational server. So we use less powerful. And also this reflects the next results. So for the whole genome sequencer, there are several base calling methods. So there is quick and fast base calling. So don't use them, but it's easy because it can be done during the sequencing, also with a lower computational resources. And you get more reliable data during the sequence instead of without any base calling during sequences. So with a slight delay of a few hours after ending the sequencing run, you have this preliminary fast base calling results. You can use them or not, but we decided also to go for the high accuracy. So therefore, we also tried doing this during the run, but to the limited resources. Yes, the run crashed. So it was too much for the hardware. Yes, it's nice to notice, but therefore we do it afterwards. And it needed up to 1 day. Yeah, the resources we have so far available are very limited, and we are in discussion with our IT department to get on the high performance cluster. But therefore, we have this high accuracy results, and therefore we can move on to varvis and set up the automated pipeline for uploading and also analyze the data in the secondary analysis step with varvis, so therefore we can for. But we also tried to run the super accuracy model. And if I ask you, maybe you want to think about them. We need 6 days for one genome sequencing dataset to get the results for the super accuracy. So there's no possible way to use this setting anymore in a clinical diagnostic way. And this is really exciting to know it now. So now we can go back to the IT department and say, okay, if you want, we have to wait 6 days for the data analysis, then you will be responsible for it. Therefore, give me the high computational power. I didn't know whether it works, but we try to do this way. For the adaptive sampling itself, you need at least to go for the fast base calling, because during the run, each molecule will be sequenced 1 second for 400 to 500 base pairs. The base calling will done, then it will be compared with your target you are interested in, and the decision is made whether it's sequenced further or it's released from the pore, and the next molecule will be sequenced. So for sequencing itself, also here the fast base calling is necessary and suitable, but afterwards also here rerunning an additional high accuracy base calling. So looking at the steps and the time frame, we want to know how this can be implemented. So as I showed you, first day for the DNA QC and the library prep is possible. Sequencing takes in 3 to 4 days, depends on the quality of the flow cell. So also therefore is not really plannable in the beginning, because you will see during sequencing whether the active pore amount will drop immediately or it really will go for 3 or 4 days sequencing. So for the IT resources, I hardly recommend following the guidelines from ONT or not doing this way like we, so buy an P2 integrated or larger instrument with a suitable hardware. So therefore, go for this recommendations. It's really needed. because it's somewhat different. For the short read sequencing, it was much more easier to upscale the analysis from several exomes to 100 exomes to genomes or something like that, only using CPU power. But here you use GPU power, and this is somewhat different. So therefore, it's possible, if you have a suitable hardware, to do this in less than a half day. But so far, this is our major pain point. But the secondary analysis, not as much easier in comparison to that, because with the guy from varvis, we talked about much. We do the automated pipeline, uploading, and it would be better to go for the analysis in parallel for the sequencing, but instead, or what we aimed here, doing this afterwards, you lose a lot of time. So the sequencing itself takes a lot of time. If you need additional, after finishing the sequencing, a lot of time for the first data analysis, and then it goes a little bit faster with the guys from varvis. But here you can lose a lot of time in it. Diagnostic settings, each hour or each day you save is an advantage. So let's come to the first results. So for the adaptive sampling, we use two panels. Let me call them panels, because the first was a commercial panel from the Oxford Nanopore technology facing the hereditary cancer diseases. And the second one was a custom panel, because we also are interested in the flexibility of this methodology on this instrument. So therefore we decided to go for a repeat expansion panel. I will show you why and what we get here. So for the hereditary cancer panel, here you can see the results from a female patient with an hereditary breast and ovarian cancer syndrome, where we identify in the SNV analysis 2 heterozygous CHEK2 variants. And in this case, we want to know whether they are in cis or in trans, because it's important for the clinical outcome. And we use this long-read sequencing technology. Also, the varvis software can phase this information, as you can see here. So it's the same phase, but two heterozygous variants are located on two different alleles. So that means they are in trans. And here directly we have additional information compared to the short read before, because it's also possible to phase something or to state whether they are in cis or in trans with short read sequencing technology. But then the two variants must lay within the up to 150 base pairs. And that's normally not the case. They are so close to each other. So if they're far away, then long read sequences have additional advantages. So here we see a second case. So it was a male guy with a suspected neurofibromatosis type 1. So therefore we identified a deletion on chromosome 17. And if we zoom in, so also the responsible NF1 gene is located in this deletion. But beside this NF1 gene, there are additional genes that are located in this deletion. So that means that here in this case, we have this typical NF1 microdeletion syndrome of 70q11.2. And it also has the typical size of 1.4 megabases. So not all of these genes are in the target region of the hereditary cancer panel. NF1 is, it's a main target. But using this adaptive sampling technology, you also get a low coverage information on the whole genome. And this information can be used also in the copy number analysis. And therefore, here identifying the breakpoints of this deletion. So let's come to our first experience with the custom repeat expansion panel. So we are doing a lot of fragment analysis for repeat expansion. So including the Fragile X, the FMR1 locus. We do a lot of for the muscular dystrophies 1 and 2, or the spinocerebellar ataxies, as well as others. So therefore we thought, okay, catching all of them in one analysis would be suitable, and then let's test them. We follow the recommendations from ONT regarding the bed file generation, the sequencing itself, and also the fragmentation of the DNA for about 8 kb. And we will see whether this works or not. So here are the first results of a female patient with suspected FMR1. So therefore, we saw in the software by using the STR analysis directly, we have two alleles. On the one allele, it's a normal one with a count of 28 repeats, so it's in the normal range. But on the others, we have an enlarged, so expanded alleles with around 122. This is an intermediate or a pre-mutation allele, and it's easily visible. Also, the information from the ST archives are here about the region. So what is the benign range, what is the intermediate range or the expanded range. And also there is an interpretation of the software, okay, where does the repeat counts lie? So therefore it was easily analyzed and identified. So here I have a male patient. And in this male patient, we have also two alleles. Okay, let's think about it. We have a normal allele. No, this is an expanded allele in the intermediate range with also around 83 repeats. And we have a really expanded to the pathogenic region with more than 200 here repeat counts. And if you look in the interpretation, so this is really an expanded pathogenic allele. But one moment, so this was a male case. So they should only one X visible. So therefore, we go for the copy number analysis and have a look on the X chromosome. And really, yes, there's only one copy of the X chromosome. On the ends, on P and Q arm, we have the pseudoautosomal region 1 and 2, and also there was one Y chromosome. It's a normal XY male. So that means that phasing or identification of these two alleles must be a mosaic in this patient. It's clear, it's objectable, these patients exist. And also this was easily identified. What we are wondering about is, are these interruptions on the second allele that reflects and it was not able to see in the prior analysis. So because they are always, we are focusing on the pathogenic CGG, and now with the long read sequencing, we see the interruptions on this loci or within the repeats, and it's known about some of the repeat or short read diseases that also the disruptions or interruptions are necessary to analyze, because sometimes the whole fragment size counts or only the pathogenic motive. So let me show you another case. So this was an additional male case where we looked for the CNBP allele, and for this disease it's known that very large repeat expansions are causative. So on the first view on this result, it looked more or less normal. But expecting the information the software gives us, they show that on both alleles, there are more than 14 repeats. So this is somewhat interesting, because why the software tells us 14 or 15? Why more than 14? And if you look here in the region that are associated with the disease, so there are very, very large expansions and also this. But the software gives us another hint. So therefore here is written down, user inspection of BAM file is required. So this is the first software for this loci that really in the first give us a hint that there could be an expansion. We tried also other algorithms and pipelines, and none of them showed us to us. But we know, because we studied this case before by Southern blot analysis, that in this case we have more than 3,500 counts of these repeats. And if we multiplicate this with the motive by 4 base pairs, so the fragment size for this is more than 12 kb. And if we are focusing on an 8 kb average fragment length, it's clear that we never will get this expansion for this. But the software gives a hint that maybe the 14 here is not the truth and there must be something else. So also this is an advantage so far. So let's switch me to the whole genome results. So this was the first male patient with a developmental delay. He could not sit, work, and speak at that times where we face him. He had seizures and a muscular hypotonia. So a few years ago, when we saw this patient to make the diagnostic first, we do a lot of analysis. we looked for fragile X, and the M1 is all negative. We make a chromosome analysis, in conventional one, and here you can see the karyotype. It looks quite normal, also in my eyes, but I'm not a cytogeneticist. So we do an array analysis, and then there was a 20 megabase duplication on chromosome 10. So, to my opinion, 20 megabases maybe could be seen in the chromosome analysis. Therefore, I asked our cytogeneticists, and they went back to the karyotype, but 2 copies of the chromosome 10 looks quite normal. But without the information that there must be something, also our cytogeneticists looked in more detail and found that this chromosome 15 looks quite different regarding the P arm. So it's an acrocentric one. Maybe it's challenging so it's okay that this was overseen in the first view, but now with this additional information, so this was carried out. And then our cytogeneticists make a FISH analysis with a whole chromosome painting for chromosome 10, and we have here the 2 normal chromosomes 10, but we have an additional signal on this chromosome. And regarding to the hint from the more detailed chromosome analysis, this could be in chromosome 15. There was also a sensory probe for chromosome 15 in green, showing here the normal chromosome 15 and here the other green signal. So this is the aberrant chromosome 15 and confirming the translocation from 10 to 15 in this case. So we choose this to get an idea of the potential of long-read sequencing. So we do the long-read sequencing, and for sure, we saw the 20 megabase duplication on chromosome 10. So therefore, we want to know whether we got more information out of them. So using the HBO term and entering to the software, as mentioned before, we can use them, and also we filtered for the HP similarity score. So this was the first top hit and also the course for this patient. But what is with the translocation? So if we are looking at the structural variants, we see that there is a translocation from chromosome 10 to an unknown contig. And there's another one from an unknown contig to chromosome 15. So maybe this is the translocation we see also in the chromosome and FISH analysis, but it's not directly visible in the software so far. But if you have a look on the data, so we used HG30H, so in HG30H for the acrocentric chromosomes, the genome reference is missing some information, and therefore this information is distributed of several contigs, and therefore it was not possible to claim this in the first. So therefore this is a hint for we need a more comprehensive genome reference. So we are looking forward that the T2T will be more completed, incorporated in public databases, and then also maybe in this varvis software in future. So, in the second case, this was a female patient with intellectual disability, with autism, and bipolar disorders. Also, therefore, we love our area analysis, and therefore we identified a duplication on chromosome 22. So this is a typo, and I hope you're active listening to my talk, and then you found it out by yourself. Following by a deletion on the same chromosome on the real telomeric end. This was also visible in the short-read exome sequencing data, but we want to know, is there more? So therefore, we use the long-read sequencing, and yes, we see both of them, the duplication as well as the deletion at similar sizes. But the question was, is this a tandem duplication or something else, something like that? And therefore, we also looked for the structural variants, but there was no entry on this chromosome. So, and here we have to state that the CNV analysis have a better resolution for the large events compared to the structural ones, because they are better with the shorter events. So maybe also with the T2T or with improvements in these algorithms, we will see this in future. So, the next case was a female patient, really young, 3 years, with a developmental delay, with speech delay, short stature, and an craniofacial dysmorphism. So also the preliminary analysis was done on the array, and we found a 4.2 megabase deletion at the distal end of chromosome 20q. And our experienced data analysis on the array then raise the hand and say, oh, this could be a ring chromosome, because there are no markers on the P, and maybe we have seen this before. This could be such an event. Therefore, we go for the chromosome analysis that was analyzed at the same time, but need a little bit longer than the array. And also, therefore, we see a ring chromosome 22. So therefore, it was confirmed, but they additionally make the FISH analysis using 2 probes on the long arm chromosome 22, the red one, and the more distal green one. And here you can see the normal X chromosome, and there the green probe is missing at the end. So this is the ring 22. And the complex, cytogenetic formula. So we also studied this patient with the long read sequencing data, and as expected, we see the 4.2 megabase deletion on the end of chromosome 22. But in this case, we have a few markers that also show a slightly decreasing intensity, but not marked by the software as deleted. But if we look on the structural variants, so there was an entry on chromosome 22, that's meaning there was an inversion, and the inversion covers the whole gray region highlighted here. So this is the remaining chromosome 22 with the ring chromosome, and the other one is the normal 22. So in this case, the structural variant caller directly identifies this, but you have to puzzle this information together and make the interpretation that this is the ring. But also here, the probes on the P arm of this acrocentric chromosome is missing in HD30H. So an advantage would be to use the T2T reference genome. So let me present another case. Also a female patient, 16 years old with seizures, attention deficit hyperactivity disorders, learning difficulties, autism, and pigmentation disorder. So also therefore, yes, we love our area analysis. So we identified a 10 megabase duplication on chromosome 11 on the short arm. But inspecting the smooth signal and the BAF, so it was shown as a mosaicism. So we saw also the duplication, but not in a mosaic status in the short read sequencing at nearly the same size. And in this case, we're doing additional MLPA analysis, and also this confirmed the mosaic status of this duplication. So, but unfortunately, no chromosome analysis was available at that time because no materials from the patient. Using the long read sequencing technologies, we also see this duplication, but in this time it's separated in 3 different duplications, where we also have here different log2 ratios, meaning that the mosaicism is maybe somewhat different. But also in this time, the structural variant caller gives no further hint where is this duplication located. It's a tandem duplication or something like that. So therefore, we got a more closer view on that, but also not all of the information. And my last patient I want to show you is this male patient, which was very early sent to us for the molecular analysis with a developmental delay, muscular hypertonia, and no other features visible at that time point. So also therefore, we went for chromosome analysis that was negative. There was, at this stage, no need for further FISH analysis, but the array identified a recurrent deletion on chromosome 15 in the Prader-Willi-Angelman critical region. So therefore, we expected maybe with the presentation of the patient, it could be an Angelman syndrome, and also in the short read exome sequencing data, it was visible, but we need an additional method. So we choose the methylation-specific MLPA as a standard method to look for the methylation on this locus, because it's an imprinting disease, and we have a different methylation between the paternal and maternal allele. So the normal MLPA confirms the deletion on this side, and the additional methylation analysis showed an hypomethylation and therefore confirmed the Angelman syndrome in this case. So using the long read sequencing technologies, it must be possible, hopefully, to solve this case in one try. So therefore, yes, we see the deletion, the typical one, also with the typical size. And now the question is, do we further need a methylation-specific MLPA? Because it's possible to do also methylation analysis and sequencing during the running cells with the long-read sequencing. But so far, I have a look on the right. So there is no feature in the varvis software so far. And maybe we can discuss this later with the varvis. So to summarize, we need more computational power. We definitely need more. So if you start, don't make the same pitfall as we do, because it was not my decision. So what our IT company, but then they not deliver when they should. So, but the whole genome sequencing worked quite well. Yeah, there's some advantage, some disadvantage we will see, but also the adaptive sampling works, but especially for the repeat expansion due to the slow size, the difficult regions to be sequenced, they need more optimization. And also we have to decide between the really short repeat and the longer repeat and the long repeats. So therefore we need additional optimization for the library preparation. The software so far do not fully exploit the potential, but it's not only the software, it's also we need a more complete reference genome. And then I think also the software at this time would be able to cover more of the structural variant, as I showed you, for example, for the acrocentric chromosomes. So at last, I want to thank my colleagues, because this work was not done only by myself. So Susan Engelberg and Sina Bialdiga was the guys who made the most experiments in the lab. Marco Rakis, our data scientist, that feasibles and set up the workflow at our side and also make the correspondation with the varvis guys for automated upload and the other resources. Then Annika Rettig, our cytogeneticist, doing the analysis on the chromosome part, and Denny Schanze was responsible not only as a medical doctor for the patients, but also for the molecular karyotyping. Also, I thank to Ben Liesfeld for the helpful and fruitful discussions about how to interpret this data, because the switch from the short read sequence to the long read sequence with the new features in the software and so on has also arised in new knowledge to us, because we know about the molecular mechanisms, but we have to switch the software and therefore also learn about the new features. And also Lena Hausdorff as product manager with helpful insights in all that. And the whole varvis team itself, because all times if you have questions, that will be addressed shortly. And also Oxford Nanopore with the tech support. We had a lot of discussions to improve the settings, the laboratory settings, as well as fragmentation or some of the other steps. And I hope you enjoyed this talk, and maybe you're interested in what's going on with this, because we had a lot of interesting cases we're discussing so far, and maybe with additional features in the software will solve directly, and maybe there will be a second talk like this in future covering the other interesting cases. Thanks a lot.